I will be unfortunately in clinic today, seeing other kids and their parents, but tomorrow I'll be there in the flesh. So happy to talk to anybody about specific issues at that time. But really, it's important to have conferences like this when parents get together and form these groups. It really helps push therapy and research forward. So just congratulations on the effort of everybody. Um, so I'm going to start just talking about, in here with the ACTA2 Alliance. I'm just going to start about ACTA2 because that's what people are here to talk about. ACTA2 is the gene that contains the information to make this protein called alpha smooth muscle actin. But I think, you know, most people who aren't in science or medicine have never heard of actin. So just want to take a step back. It seems like, it probably seems like everything we know about these conditions came from the last couple of years, but it's really actin research is really old, and it's almost 80 years old now. And that was because, back a long time ago, like for instance, in the 1940s, what I'm showing you here, people were really interested in muscle biology because muscle was kind of exciting and it was this thing that moved in the body. And it was really accessible to researchers in the period and the techniques they had. So this group at the University of Szeged in Hungary, um, the actually the folks who did the research were Bruno Straub and Ileona Banga, work in the laboratory of Albert Gorgieri. So they actually were the first group that discovered actin itself. And they discovered, that there were different forms of actin and that it could polymerize and that it was driven energetically by this molecule called ATP. And the way they did that, this was really clever back in those days because you didn't have access to the technology that we have now. They took rabbit skeletal muscles, so just the regular muscle from a rabbit, and ground it up, and then made extracts. And they found that they could extract this protein reliably with potassium chloride, and they would get this pure protein that they could then bring out a solution and make these thin films, like the one over on the right with the red arrows. And they called it myosin, which just means, protein from muscle, but they didn't know what it did. And so they were looking for basically other molecules that made it move. So they took the extract that didn't have the myosin in it anymore, and they purified it in lots of different ways. And eventually they found this extract that they could pour on the myosin, and it would cause it to contract really. These films would zoom up and contract. And they described it as a violent contraction. And eventually, this was the first demonstration of this protein called that they named actin. This was in 1942. So, what is it? Actin, this is a picture of an actin monomer. So, it's just a protein, like other proteins in the cell. We have, tens of thousands of different proteins in humans. This particular one has the ability to form filaments. Um, and they form long filaments, as shown in this illustration, and they align with that protein we talked about before, myosin. And myosin has a feature that it has little motors on it, and those motors can grab onto actin and make it slide. And so this is what creates the motion or the contraction in muscle, is the interaction between actin and myosin. And skeletal muscle, these are very small, and you get them together in the sarcomere, and then you bundle more together, and you bundle more together, and you bundle more together, and eventually you have, a muscle like shown here, like a biceps muscle. So this is how the muscles that you can voluntarily control work and how they're based on actin-myosin interactions. What about smooth muscle? It's different, but very similar in that instead of having these long alignments of actin myosin, they have more of a network across the cells. And so smooth muscle containing organs like arteries or intestines have um, have lots of smooth muscle cells, and their actin-myosin networks are distributed across the cell and cause the whole cell to contract, and eventually the whole organ to contract. Now, there's different types of actin. So there's actually in humans 6 isoforms. And some of them are present in all cells, like beta actin and gamma cytoactin. They're present in every cell in the body. And then there's some specialized ones that are present, just within certain muscle groups: skeletal alpha actin, skeletal cardiac actin, and there's two smooth muscle actins. One is, of course, alpha smooth muscle actin, and the other is gamma smooth muscle actin. And all these different actins, they don't vary by a whole lot, mostly from this little N-terminal region that's shown up here. But they do different things in the cell, and they are expressed in different types of cells. So for instance, here's an example in arteries on the right where you have alpha smooth muscle actin and gamma smooth muscle actin to some extent. In veins, we see alpha smooth muscle actin, but a lot more gamma smooth muscle actin. The exact reason for all these combinations aren't always clear, but it's what we observe. And each individual isoform actually is encoded by a different gene. So the genetic information that that's in your genome, in the DNA, that encodes for these different actin proteins are different. So each of these actin isoforms has its own gene with its own associated problems if dysfunctional. So for instance, the two actin genes that are expressed across the entire body, as you would expect, if they're dysfunctional, they cause a disorder that's present across the entire body. This is called Barrett-Winter syndrome. And these children can have dysfunction in lots of different body systems. So anywhere from, brain malformations, they have a distinctive facial type. They can have neurovascular dysfunction and ocular issues. And when you look at the cells, which is the bottom row, the actin fibers in these cells isolated from patients have different features and abnormal features. And not surprisingly, the other isoforms that are expressed more in specific muscle tissues end up causing specific muscle problems. So ACTA1 is a skeletal muscle alpha actin, and it causes a skeletal muscle dystrophy. Similar but not the same as a condition like Duchenne's muscular dystrophy, which you've probably heard of. But it can cause problems with skeletal muscle. The cardiac alpha smooth muscle, or excuse me, alpha actin causes cardiomyopathy. So dysfunction of the heart, most commonly seen as dilated cardiomyopathy with eventual heart failure. And then the other smooth muscle actin is ACTG2. It's generally called a visceral gamma actin, but it causes bowel dysfunction because it's mostly expressed in the intestine. So when you have mutations in the gene ACTG2, you get dysfunction really of the intestine, and then stenoses and dilation of the viscera or the intestines. Okay, so just to give you a sense, the reason I'm going through this is to give you the sense that ACTA2 is, not the only actin-related condition, and so these things are being researched and looked at by multiple people in different fields. But we're more specifically, of course, here to talk about ACTA2. ACTA2 encodes smooth muscle alpha actin, and it's present really in every tissue because every tissue, all the different parts of the body, have some smooth muscle. But some organs like the arteries and the intestines, for instance, their function really depends on smooth muscle. So as I mentioned before, when a smooth muscle cell contracts, the whole cell contracts. And when those cells are linked together, it can cause, it basically allows the organ to contract or relax. So this is an example on the bottom right where you have a regular artery in the middle, and there's a stimulus so that the smooth muscle contracts and the artery gets narrower. Or there's a stimulus to let the artery relax, and so the artery gets larger. And that allows the body in very specific ways to regulate blood flow. So when you're exercising, you need to put more blood to your muscles to get your skeletal muscles working, or when you're digesting, you need more blood to go to your intestines. That this is the function of the smooth muscle contraction. But the story of dysfunction in actin 2 actually came about 20 years ago with the first description of disorders that were caused by gene changes or mutations in actin 2. And this was described by Dongchen Gao and Diana Milowitz in Texas, where they described mutations in the alpha smooth muscle actin gene led to this disorder called thoracic aortic aneurysm. And aneurysm, there's different types of aneurysms throughout the body, but TAA, or thoracic aortic aneurysm, most commonly occurs right above the heart in the part of the aorta that's called the ascending aorta. And they noted large families that had multiple people that were affected by TAA, and then the danger in thoracic aneurysm is a tear, or what we call a dissection. And so these families had both this finding of aneurysm, but also dissection, and identified gene changes in the actin-2 gene that cause that. And they're passing through the family, mediating the problem. What they noted, when you look at the actin-2 gene and the protein that's created from it, the alpha smooth muscle actin protein, is that the gene changes, the actual amino acid changes that are caused by the mutations are spread throughout the protein. And there's some recurrent ones, but they can be in lots of different places. The proteins, about 380, 370 amino acids, and you can find these changes along the way. They tend to be missense changes, which means there's a spelling error, like in one amino acid, and they mediate that aneurysm. But what Dr. Milowitz and her colleagues noted was there was one place in the actin-2 gene that caused a really different syndrome, and that was at arginine 179. And that caused a much more aggressive aneurysm condition, but it also caused other findings, not just in the vascular system, but in smooth muscle really throughout the body. And they named that multisystem smooth muscle dysfunction syndrome. And they described that in 2010 in a series of, in a case series of about 5 patients who all had de novo gene changes in the actin-2 gene at arginine 179. And that de novo just means they're the first person in their family. They didn't inherit it from mom or dad. And so in these patients, they described the aortic disease, but also neurovascular problems and pulmonary problems, bowel problems, and urinary, so bladder and kidney dysfunction. So really a multi-system problem with smooth muscle. And so now this is the way how we think about MSMDS, is that it really has symptoms throughout the body that are relevant or caused by smooth muscle cell dysfunction. So in the ocular system, mydriasis or dilation of the pupil, in congenital heart disease like patent ductus arteriosus or aortopulmonary windows, pulmonary hypertension, and of course aneurysm dissection. In the cardiovascular system, of course, we see more than that, though. We see low blood pressure and dysfunction of the autonomic functions of the body, that the way your body controls your blood flow without you thinking about it. Um, in the bowel, of course, we can see dysmotility and malrotation, prune belly disease, seen gallbladder stones because your gallbladder empties by smooth muscle. Lung disease that's characterized by emphysema and bladder and bowel dilation and dysfunction. And of course, one of the characteristics, really the unique characteristics of the condition is a cerebrovascular disease, where the blood vessels in the brain are different. Morphologically, just from sort of the pictures we can take, they show proximal dilation of the carotid, but then tapering and stenosis of the distal internal carotid. Overall, there's loss of tortuosity. In most of the body, tortuosity is abnormal, but in the brain, it's quite normal. The arteries are tortuous or twisty, and in MSMDS, they're straightened, and they have a thinner caliber than they should. And then, of course, what as a cardiologist, I'm most familiar with is the aortic condition, which is dilation of different segments of the aorta that we need to, address most commonly or with surgery if they get too large. So mutations or gene changes at arginine 179 are the most common cause of MSMDS, but one of the tasks of this talk was to talk about mutations in other genes and could they cause MSMDS. The answer is yes in limited cases. So there's been a couple patients reported that have had identical neurovascular disease to patients with ACTA2 mediated MSMDS, but have had gene changes in MYH11. A couple of these patients also had, disease outside of the neurovasculature, like patent ductus and thoracic aortic dissection. Now, MYH11 um, is familiar in smooth muscle because, as I mentioned before, ACTA2, is the alpha smooth muscle actin and can make these filaments in smooth muscle. MYH11 actually encodes smooth muscle myosin. So it makes some sense that gene changes in MYH11 could cause a condition quite similar to MSMDS. And so we see mutations in MYH11 causing aneurysm and dissection, and then also some aspects of MSMDS, like the neurovascular disease. There's one more that we characterize. We met an 18-year-old at MGH who had multiple signs of MSMDS, the cerebrovascular disease, prune belly syndrome, gastrointestinal dysmotility, such that he had needed to be on, IV feeding, bladder dysfunction, and pupillary dysfunction. But despite all these signs, he had no changes in his ACTA2 or MYH11 genes. And so with Dr. Sweetser in the undiagnosed disease clinic and Dr. Mussolino, we underwent characterization of his genome, thinking that this must be also an undescribed cause of MSMDS. He had the characteristic neurovascular disease that was progressive and caused that same characteristic dilation of the proximal carotid and the narrowing of the distal carotid. And what we found was a gene change in a microRNA. MicroRNAs are genes, but they don't make proteins. They just make these small little RNAs that can go and work inside the cells and in different tissues, and they act by regulating other genes. MicroRNA 145 was really famous already to smooth muscle biologists because it had been identified as a microRNA that regulates smooth muscle cell function. And the gene change in this child landed in a very specific part of the microRNA 145 gene. It's a part that's required for its basic function. It's this track of 7 nucleotides that are absolutely conserved in evolution. And there was a gene change in this particular individual. And what that caused was his microRNA 145 to not be functional. And not only was it not functional, it was dominantly affecting the other allele. And so as a result, and you can see in this modeling experiment where we put in the mutant, we made a mutant that matched his, he was deficient in smooth muscle actin um, expression, and so it's not too surprising that he ended up having a medical disease that was similar to MSMDS, and so we reported that a couple of years ago. So, what to conclude? Just to wrap it up. So, ACTA2 R179 is the most common cause of MSMDS, and some of the other ACTA2 variants can cause different parts of MSMDS, but the fulminant condition is really seems to be most common, if not unique, in R179 mutations. And that's biochemically, it's because it's a very vulnerable amino acid that causes really a lot of dysfunction in the ACTA2 protein, but also is able to dominantly affect the other copy that's normal. We also see MSMDS-like conditions in MYH11 and in this microRNA. So overall, these loss of function changes are thought to really just destabilize smooth muscle cell dysfunction. The molecular details are somewhat different between them, but the result is that you have a smooth muscle dystrophy, or where basically smooth muscle is not doing what it's supposed to do, and then the medical consequences are the function. So just thank everybody. This is our gene therapy team for pediatric vasculopathy. This was from the last ACTA2 conference, and all the kids that came and parents, thanks, and thanks for coming again this year, and happy to talk to y'all in person tomorrow. So happy to take any questions about this.
Questions? Oh, we do have one question.
Just one simple one. How do they know it's de novo if the parents have never been tested? Hi, good morning. How do the physicians know it's de novo if the parents were never tested?
Yes. So there's different ways of knowing de novo. So you can just test the parents, which is done sometimes from a research standpoint. But if the disorder is severe enough, we basically intuit that the parents don't have the gene change. There's an exception, though, that some people can have can be mosaic, or they have a gene change in one organ that they don't have in another organ. And occasionally that can happen in the germ cells, like the testes or the ova. And therefore, we occasionally can see a disorder even though the parents don't have the disease, but a parent could pass it on more than once. That's unique and rare. Most of the time, it's just when the disease is severe enough in a child, we are assuming that the parents don't have that gene change. But we can formally test it as well. It's very easy to test.
I have another question. It's kind of two questions. One, is there any way to explain all of that in layman's terms?
Which part?
And,
All of it.
and two, as the father, I had a rare disorder as a child. I had Kawasaki syndrome. Is there any relation?
So yeah, so Kawasaki's disease is not as rare as we once thought it was. We have children with Kawasaki's disease in the hospital all the time. It's an inflammatory disease, kind of like an autoimmune disease, and it seems to be set off by a virus. And then you get an inflammatory attack of your arteries. And so it's really a vasculitis. It's not related to this, and its genetics are really quite different. Um,
One more question.
Thanks, Mark. Mark, you said the microRNA mutation was dominant, but could you
biochem Biochemically.
exogenous?
So we think it's dominant both from the patient phenotype, um, because there are loss of that allele without the phenotype, and then biochemically in humans, and then biochemically, when we tested it, it seemed to have more than a 50% effect on alpha smooth muscle actin function.
Questions? All right, Dr. Lindsay, thank you very much.
Thanks all. I'll see you tomorrow. Happy to talk to everybody then.